Hello everyone, we are here to discuss the gene therapy. Gene therapy is a potential method to either treat or cure genetic-related human illness. Human gene therapy was actually first realized in 1971 when the first recombinant DNA experiments were planned. It can be simply viewed as the insertion of foreign DNA into a patient's tissue that hoped to successfully eradicate the targeted disease. It was actually inspired by the success of recombinant DNA technology which occurred over the last 20 years. Without a doubt, gene therapy is the most promising yet possibly unfavorable medical field being studied. Fundamentals of gene therapy. 1. Vector delivery systems. Gene delivery relies on vectors, which are vehicles used to transport therapeutic genes into target cells. Viral vectors or non-viral vectors are commonly employed to carry the desired genetic material into the cells. 2. Non-viral vectors. Non-viral vectors include liposomes, nanoparticles, naked DNA, or artificial chromosomes. These carriers can transport genetic material into cells without the use of viruses, offering advantages such as reduced immune response and toxicity. 3. Gene editing. Gene editing tools like CRISPR, Cas9, TALENs, transcription activator like effector nucleases, or zinc finger nucleases enable precise modification of the DNA sequence within a cell. These tools can target specific genes to correct mutations, replace defective genes, or modify gene expression. 4. Safety and efficacy. Preclinical studies and rigorous testing are used to evaluate potential risks such as genetic alterations, immune responses, or adverse reactions. 5. Regulation and ethical considerations. Gene therapy research adheres to rigorous regulations to uphold ethical standards, patient safety, and responsible practices, ensuring informed consent, privacy, and equitable treatment access.
Techniques used in gene therapy. 1. Viral vectors. 2. Non-viral vectors. 3. Gene editing. 4. Inducible promoters. 1. Viral vectors. These vectors, commonly used for gene delivery, are modified to carry therapeutic genes and efficiently enter target cells to deliver genetic material. 2. Non-viral vectors. Liposomes, nanoparticles, naked DNA, or artificial chromosomes are carriers that transport genetic material into cells without viruses, offering reduced immune response and toxicity. 3. Gene editing. Gene editing tools like CRISPR-Cas9, TALENs, and zinc finger nucleases enable precise DNA sequence modification in cells, targeting specific genes for mutation correction, gene replacement, or expression modification. 4. Inducible promoters. Promoters are DNA sequences that regulate gene expression. Types of gene therapy and their application and implication.
1. Somatic cell gene therapy. 2. Germline gene therapy.
1. Somatic cell gene therapy. Application and implications.
2. Germline gene therapy application and implications. Now, we will explore the current applications and potential of gene therapy in some different aspects. Gene therapy aims to correct underlying genetic diseases by introducing or modifying genes in patient cells. For such diseases as cystic fibrosis, muscular dystrophy, and sickle cell anemia, this approach holds promise. Gene therapy seeks to alleviate symptoms and improve quality of life in affected individuals by delivering functional copies of the defective gene or modifying gene. It is being investigated as a way of enhancing the immune system's responses to cancer cells within an area of cancer treatment. CAR-T cell therapy, in which the patient's immune cells are modified with a view to better recognizing and destroying cancer cells, is one of the most notable approaches. In some forms of leukemia and lymphoma, this has proven to be very successful. Moreover, gene therapy may be able to extend beyond these current applications. As regards other genetic disorders, such as Huntington's and hemophilia, studies are ongoing to investigate the use of the medicine. In addition, gene therapy has the potential in areas such as heart disease, neurodegenerative conditions, and infectious diseases.
However, the field of gene therapy is still in its infancy and faced with a number of challenges. Ongoing areas of attention are to ensure the safety and durability of therapeutic interventions, as well as address immune responses and refine delivery methods for targeting specific cells or tissues. Gene therapy is currently applicable to the treatment of genetic disorders and certain types of cancer. Its potential can be extended to a wide range of medical conditions, and ongoing research is intended to unlock its full capacity. It holds potential for revolutionizing how we approach and treat different diseases as advances in this field continue.
Hey. Yes. I have a question. Miss Althea Romano. What is it? Florence. Do you think that using gene therapy should be allowed? I mean, in the context of gene therapy, it is all about modification the genes of the people for changing who they are. Do you think should we allow it? Well, in terms of gene therapy, I do know that it was allowed to do the therapy. But on the other hand, there are some ethical, social and regulatory use concern about it. Wow. I never knew about the aspects of those. What about it? Well, let me tell you about it.
Ethical aspects of gene therapy. 1. Informed consent. Ensuring individuals understand the risk, benefits, and potential long-term consequences of gene therapy is crucial for obtaining informed consent. 2. Germline editing. The ethical implications of editing genes in embryos or germline editing involve considerations of altering future generations and the need for responsible use. 3. Equity and access. Addressing concerns about equitable access to gene therapies, avoiding disparities in availability and affordability across different socioeconomic groups. Social aspects of gene therapy. 1. Stigmatization. The potential for gene therapy to inadvertently contribute the stigmatization of individuals with genetic condition and how to mitigate such effects. We can encourage viewers to critically evaluate the ethical dimensions of gene therapy by promoting informed dialogue. And this promoting informed dialogue is emphasized the importance of informed and open discussions, urging viewers to seek diverse perspectives to gain a comprehensive understanding of the ethical considerations involved in gene therapy. Second, engaging with experts in biotics and related fields to deepen their understanding and challenge preconceptions, fostering a well-rounded perspective. Third, public perception influence. Highlight the impact of public perception on the development and acceptance of gene therapy, encouraging viewers to contribute to shaping a positive narrative based on well-informed and ethical perspective. And the last one is community engagement. Encourage viewers to engage with their communities to discuss the ethical implications of gene therapy, fostering a grassroots understanding and awareness that can contribute to responsible decision-making. By actively participating in these discussions and critically evaluating the ethical dimensions of gene therapy, viewers can play a pivotal role in shaping the responsible development, regulation, and societal integration of this transformative technology.
It seems that gene therapy does have its aspects and concern about its regulatory use. And it says right here in the article that it is a newly developed approach that emerged at the end of 20th century. But way back in the 19th century, There was a concern because during the experimental procedure they had a patient and it died. But there are some cases that the therapy was successful. But according to its ethical problem, gene therapy attracted a public controversy because of unexpected side effects, which, in its utmost extent, have taken people's life away. Wow! I never knew about this. Thank you, Ms. Alva. Now we know the aspects of social, ethical and regulatory use of gene therapy. See, I told you so.